Thanks.
I think this meeting is a good testament to the fact that the DMS-style experiments have gotten much more popular in recent years. And as that has happened, different labs have developed a number of distinct methods, both published and unpublished, to tackle some of the technological hurdles involved in doing these kind of experiments. And especially at the level of library construction, there are a number of different strategies that people will use. So my goal here is to give an overview of some of the staple strategies that's used in our lab and nearby labs to generically tackle a lot of questions that can be tackled by deep mutational scanning. So before I get into it, I just want to make sure we're all on the same page in terms of terminology, because there's a lot of jargon that gets thrown around. So in general, when we're constructing a library, we've got a gene or some sort of sequence that we care about, and we're introducing mutations to that gene. And then frequently people will add a barcode that is a separate sequence that reports on what particular mutation you care about. In addition, your gene can contain multiple mutations, and we call the full gene containing those mutations variants. So before I get into different types of libraries that you could construct, I want to go over some guiding principles that I think are not obvious that sort of apply to all of these strategies. The first is that you should really have a good idea of what your sequencing/analysis strategy is going to be before you even think about how you want to construct your library. So in general, you don't want to build your library and then think about how am I going to analyze this. You want to already have a really good idea of what sort of statistics and how you're going to process your reads and all of that, and then tailor your library construction to that analytical approach. Second off, make the hard parts easy. So library construction is a hard enough step in its own right. So you don't need to necessarily do all the construction in the final vector where you're going to express your protein in some particular system. Do everything on an easy, high copy bacterial plasmid. And then once you've got the library constructed, you can port it into whatever expression system or whatever you happen to need. Third is that as you're going through your library construction, spot checking by Sanger sequencing and or growth curves, if that's a possibility, is a great idea. So you don't need to be deep sequencing your library after every construction step. You can just pick 20 or 30 colonies and look at what your mutation distribution is. And this can save you a lot of time and money if the inverse PCR doesn't happen to work the first time, which I think is pretty typical. Fourth, you want your library to contain variants that you know are going to be functional or that you know are going to be dead prior to going into selection. And in many cases, if you're just working on ordinary protein, you can just use wild type synonymous variants and nonsense variants, and that'll give you a good idea. But maybe wild type is not active in the particular condition that you're assaying in or something like that. In that case, it's a good idea to spike variants in at 1% or something like that, that you already know the function of, so that as you do your selection, you have a baseline for what you think should be happening. And then last, you want to keep track of your wild type abundance and or library complexity with a knowledge of how your selection is going to affect your library. So in general, having a wild type abundance of 1 to 10% is pretty good. If you had, say, a wild type abundance of 50%, you might think, well, okay, I can sequence twice as much and it's not a big deal. And that's true enough for the input, but you should consider what will happen after your selection. And if after your selection you have 95% wild type, that can really cause problems in terms of sequencing. So you should think about getting your wild type abundance reasonably low. I'm adding library complexity in there because in some cases you might expect 1 or 2 variants to totally sweep your library. And if that's the case, you should keep that in mind as you're constructing it. All right, and then I also briefly want to talk about barcoding, which I alluded to earlier in the terminology slide. But it's common for people to add barcodes to their libraries, although not strictly required. And the way barcodes work is once you've added your barcode, you would then amplify the gene and the barcode together and do what people sometimes call subassembly or just assembly, using relatively long reads to sequence both your barcode and the variant together, and then create a map that links your barcode to your different variants. And then in a separate sequencing reaction, you can just amplify your barcodes without the coding region, and then use that to determine frequency. And then you can use your barcodes as a sort of key to then link together your variants to the frequency. And the motivation for doing this is generally that you don't want to be sequencing your coding region over and over again because it might be too long to fit on a 75 base pair Illumina read, or you might be worried that you've got too high a sequencing error rate relative to your mutation rate. So this can solve a lot of those issues at the cost of being an extra step that you have to do during your library construction process. All right, so that sort of covers the commonalities between different library construction methods. So I want to go into some of the different ones that we use. And I've got 4 here. I'm hoping you'll indulge my desire to make metaphors. So error-prone PCR is, I'm referring it to here as a tin shed, which might sound a little bit derogatory. And it's true that in a certain sense, error-prone PCR libraries are frequently lower quality than libraries generated by other methods. But the flip side is that they're cheap to build. You can construct them fairly quickly. In addition, they're pretty scalable. So you can make a really giant one, a really tiny one, and it's pretty amenable to length flexibility. Doped-Oligo methods can be really useful. I'm referring it here as the kit shed or like a tough shed. They're generally higher quality than, for example, error-prone PCR libraries, but the major restriction is on size. You can't make them too large. And then inverse PCR and one-pot based methods here are overall accomplishing the same goal in terms of the type of library that you're trying to construct. They're just doing it through slightly different ways. Where in inverse PCR, you sort of have to lay it out brick by brick, codon by codon, versus one pot, you sort of mix everything together and then make it in one go. And I'll go through each of these individually. All right, so in error-prone PCR, your mutations arise during replication errors that are introduced by an error-prone polymerase. For example, we use Mutazyme 2, which I believe is an Agilent product, but there's a number of different ones. And so you do PCR by melting your template strand, and then as you amplify it, you get stochastic errors in the products. And as you go on, you get more and more errors. And what is sort of depicted here is that you'll end up with a distribution of errors where some molecules are going to contain no mutations, and some molecules are going to contain many mutations, and then you'll have a distribution in between there. So, you know, should I be doing error-prone PCR? And in many cases, the answer is yes, especially if the time and money of the library construction is more important to you than totally maximizing your coverage. In particular, it's also very amenable for long templates. And I would say, in general, it's nice if you're thinking, I don't want to barcode this library. I don't want to, you know, take the extra time and effort to do that. Or maybe I've got a really long template and I don't want to get really long sequencing reads. In particular, it's good if you want to pull out like winners from a selection, get a few variants that you really care about, and then go back and validate those after the fact. It can be a really useful way to do that. And generally, you don't want to do error-prone PCR if you need high confidence for individual mutations without validating them afterwards. Or if you care about epistasis, it can be an issue because there's generally a wide spread of how many mutations are on a given molecule. Last, if you care sort of about the protein biochemistry side of it, and you want to have each codon substitution, you're not going to get that out of inverse PCR. You're going to get the single DNA level mutations primarily. All right, so as an example, I did error-prone PCR on a protein that I care about, which is a phage receptor. And the idea of this was to get a number of mutations in that receptor that confer resistance to the phage that I study. So did a sort of generic deep mutational scanning strategy where made a library with error-prone PCR, selected in 2 different conditions, and then pulled out the variants that I thought looked pretty good in terms of being phage resistant, but still being able to transport nutrients, which is what this protein does natively, and then validated some of those mutations and used them for some phage experiments. And it was pretty, this was a pretty useful approach for doing that. So did raw sequencing of Nextera fragments of the coding sequence, which is shown on the left, and you just get some sequencing depth that you can raise up by just sequencing more. The correlation between replicates was not particularly great, getting a correlation of like 0.5, R equals 0.5 between replicates. And that's not particularly surprising in this case for somewhat subtle reasons that I can go into if people want to know. But even though, you know, the library quality, the coverage wasn't great, we were able to pull out 9 mutations that do exactly what we wanted them to do. So this was a pretty useful strategy for what I needed in this case. In a fairly similar example, a postdoc in our lab, Ben Branson, took a similar approach to try to find variants of this protein called EPSP synthase that are resistant to glyphosate. So this is the protein that causes Roundup resistance in like Monsanto corn, for instance. But they wanted to find variants for a different EPSP synthase that comes from cassava. So they expressed it in E. coli, did error-prone PCR across the whole coding sequence, and then similarly fragmented with Nextera and sequenced deeply across the whole coding sequence, and pulled out several variants that gave the resistance phenotype that they wanted. So very similar to the previous one, this was a good strategy for pulling out a reasonable number of variants that have the phenotype that you want, and not necessarily for comprehensively scoring every possible mutation. All right. So I'm going to switch over now to talk about oligo-based methods. And all of these start with ordering a number of oligos from a company like IDT or Twist. And then once you've ordered the oligo, incorporating it into your template in some way. And so you can order mutations in oligos in a number of categorically distinct ways. So doped oligos on the left there would be if you told IDT at each position, I don't want you to make it 100% the wild type base. I want you to make it 97% wild type base, and then 1% for each of the 3 remaining bases. And you do that across the whole region that you want to mutate. And so they send you 1 tube, and it's got a pool of an absurdly large number of molecules that are all different. And then typically, if you do this on a reasonably short region, you've got common adapter ends on the ends of your oligos, and you just gibset it into your template, and that's a fairly easy step. Alternatively, you can do user-defined mutagenesis by ordering an array, and then you don't have to just take random mutations. You can take mutations that you particularly care about. Maybe they are orthologs or something like that. And frequently people will also put on common adapter arms so that you can sort of gibson it into your template as you need. I think the most sort of generic form of deep mutational scanning, though, is doing this codon by codon mutagenesis, where you replace each codon with an NNK or an NNS or an NNN. And so in this case, you would order several different oligos, so 1 oligo for each codon that you want to change. And then every position is wild type in that primer except for the particular codon, which is randomized. And then similarly, you can do a combinatorial version of this, where instead of going across 100 codons or something like that, you pick maybe 4 codons or something in that ballpark. and then have them be totally randomized in combination. So you're not going to get broad coverage of your protein, but you will get all the combinations. All right. So these are somewhat more complicated strategies in terms of incorporating them into your template, because you're not going to have common arms because you've got a tile across your entire coding sequence. So a few methods have been developed to get these into your template. I'm going to talk a little bit about inverse PCR and one-pot methods. But first, talk about Doped-Oligo a little bit. Doped-Oligo strategies are pretty nice for getting a library with a lot of variance and relatively low bias. The main restriction is on length. So there's only so long you can make oligos, but even if you can make a very long oligo, the shorter length helps you get multiple mutations within the same codon, which is important if you want high coverage. They're also really great for looking at epistasis because you can really nicely tune the number of mutations per molecule. They're not great at covering all of the possible codon substitutions, especially if they require 3 SNPs. And in general, oligosynthesis is indel prone. So one of the grad students in our lab, or during his dissertation work, although he's a postdoc now, Mike, did a deep mutational scan of a protein called sterol-12 using Doped-Oligo. And so at the mutation frequencies he used, which was, I believe, 97.5% wild type, over 100 bases, you get about 9% wild type. And then you can look at the coverage for singles, doubles, and triples. And basically what you see is that it's very easy to cover all of the single mutations. It's reasonable to cover all of the double mutations, but covering all of the triple mutations is relatively challenging, and that's going to go up for quadruple mutations and such and such. And additionally, that's for this 99 base per template, but those numbers start looking a lot worse as you go longer than 99. However, this library was pretty high quality, and so he was able to get good scores with reasonably high coverage across this area, and additionally did some nice epistasis analysis because you get really good coverage of the double mutations here, and so you can look at position by position effects quite well. All right, inverse PCR based library construction is a relatively straightforward way to make NNK or similar type codon substitutions. The idea here is you order 2 primers for each codon that point outwards from each other and touch tail to tail. And the NNK comes on the very 5' end of one of those primers. So you just amplify and then religate, and now you've got your NNK substitution. This is obviously just for one codon, so you've got to do this for every codon that you care about. We had an undergrad in the lab use this strategy to make a library of a peptide antibiotic called Klebsidin. And one of the reasons why we used this approach as opposed to other approaches is there's only 19 codons in here, so you've just got to use 19 tubes. And it worked pretty well. You got good coverage of the library. Obviously, if you saw, for example, Clara's talk yesterday, you can use this approach to study much, much longer proteins. It just involves doing more PCRs. All right, and then last, I'm going to talk about one-pot methods, which do more or less the same thing, but in a single tube. And there's a few different approaches for this that have come out that I'm not going to go to in detail, but I'll refer you to these papers. But in general, they all follow the same strategy, which is you take your template DNA, you make it single-stranded somehow, you anneal your pool of oligos to it, and you use that to synthesize the second strand, and then you degrade the first strand, and you resynthesize that with another primer that doesn't have a mutation on it. So I was able to use this strategy to study a phage protein called GPJ. And so the basic strategy was to design oligos, do nicking mutagenesis, and then Sanger sequence a bunch of them and see what I got. I saw that my library was about 30% wild type, which was higher than I wanted. So I actually spent a while trying to optimize this strategy to reduce the wild type abundance, which, you know, was a lot of just sort of playing around with enzyme concentrations and stuff. But what ended up sort of solving all my problems was just taking that library and running it through another round of mutagenesis using the same protocol. And so that got my wild type down to about 10%, and that seemed reasonable. And from there, barcoded and then cloned into phage. And you can see got pretty high coverage of the library, about 93%, and it's pretty replicable and everything, largely because it's barcoded to about 10 barcodes per variant. All right, so should you do one of these methods? In general, if you want high coverage of protein level single mutations, this is the way to go. And it can work for a pretty flexible range of templates that you might care about that are sort of in the gene length range. In general, I think inverse PCR is probably the smarter way to go if you want to be sure that this is going to work the first time you do it. If you want to do this a bunch of times in your lab, it might be worth establishing a one-pot protocol in your lab that you can use over and over again. And then the only real reasons to not use these methods are if for some reason you really don't want a barcode, or if an easier method like Doped-Oligo is sufficient for your needs. All right, so just to wrap up, I want to emphasize that there's room for creativity in these methods. So I don't think that we've fully saturated the ways to build libraries, and you should, if you've got a selection strategy or something that demands a different library construction method, you should play around with these and maybe try to figure out something unique. In general, you should figure out what you want to learn and how you want to analyze your data, and then go back and plan your construction rather than going the other way around. And some of these methods are a little bit intimidating, especially I think the one-pot methods, but they're definitely doable, and they're built for basically the people in this room. So with that, I'm happy to take any questions.